Well, hello again, Syma. Time for another edition of Science of the Times podcast, and hello, listeners. So
Hi, hi
it was good.
everyone.
It was good to see you, Syma, the other day at an Senior. Do you want to try and explain what that is?
Yeah, it was lovely to catch up with you and Sonia. So Insignia is I guess it's the garden party that happens after the honorary awards, the degrees are awarded, which is done in the Sheldonian. It's a fancy ceremony, and then the garden party this year was at Merton College. Did you go to the ceremony
I
itself?
didn't. I have been in the past. I didn't this year. And being Oxford, of course, it's entirely in Latin,
That's right.
so I didn't. But there was some well-known recipients of honorary degrees, including Billie Jean King. Did you see Billie Jean?
No, I was devastated. I didn't get a chance to see her. Sort of, yeah, a real hero, hero of mine. But I saw that lovely photos. She was there and she seemed to have had a great time. So that was wonderful.
No, I hope so. I hope so. So I didn't get to see her either,
Yeah.
but there were a few others. There was a well-known dancer was there.
Carlos Acosta. Yeah, yeah,
Yeah, and
absolutely.
yeah.
Um,
So he made me realize I need to go to the gym a little bit more.
and take up ballet dancing. I can see you doing that, Tim.
Clearly, I mean, I've always known that ballet dancers are incredibly fit, but yeah, it's
Yeah.
no. So I did see him. I didn't
Cool.
talk to him, but it was good to see him.
And so ne next week, Tim, I think you're headed to London. Do you want to tell our listeners about that?
I do.
Incredibly
It's
exciting.
a great honour, but I've been elected a fellow of the Royal Society, so that's the world's oldest scientific academy, and it's lovely because you're sort of you're nominated by your peers, and they ask for letters. So no, it's a really great honour, and I have to go up to London to the Royal Society where there's a day of talks and then there's a sort of nice meal. And then I have to sign this ancient book made of vellum with a special pen. It used to be a goose quill, I believe, but there's Newton is kind of one of the early signatures, for example. So yeah, it's kind of exciting. I mean, it's a fantastic thing to have happened. I'm absolutely delighted, but also slightly bemused that it happened to me, sort of thing. So.
No, it's thoroughly well deserved. I think it's a great honor, and I'm chuffed to bits for you. I'm just a little bit worried though that you might pour a glass of wine over this ancient book.
I don't think
Absolutely
there'll be any wine near the book, but there might
excellent.
be an inkwell or an ink bottle. So yeah, I'm gonna have to be. Apparently, there is someone on standing there with special blotting paper because it's onto vellum, so animal leather. Apparently, you can mop it up if you're quickly
Yeah,
enough. But
excellent.
yeah, I just have to make sure I can spell my name right, I guess.
Yes. I mean, Tim, even you should be able to manage three letters.
Yeah,
But
I know, but imagine how stressful it's going to be. I might like, shit, I forgot my name, might sign something else. But
anyway, we've got an absolutely exciting guest lined up for today, haven't we, Tim?
we have. So Isabel Smallegange, I think I've said her
Yes.
name right. I've known her for several years actually. We were at Imperial at the same time. But she's now at Newcastle and she does really interesting stuff. We haven't had anyone on like this before because she works on animals, but in the laboratory. And these are tiny, tiny, tiny little like eight-legged beasts. So, but we should let her explain, I guess, what's going on.
Yeah, absolutely. Let's get her on.
Yeah. Okay.
Okay, so hello, Isabel. Lovely to have you with us.
Thank you. Thanks for having me. Nice to
You're
see
you're
you.
welcome. So, would you like to briefly introduce yourself to our viewers and listeners?
Yes, so yeah, hi everyone. Um, so I'm Isabel Smallegange. I'm a population biologist. Yeah, and I one of the main questions that sort of drives my interest is why do we see differences between individuals and how they look and how they behave, and how does that impact the dynamics of population? So my research sits a bit at the intersection of ecology, evolution, but also development, which I think we're going to talk about a lot. And and I study tiny bulb mites, amongst others, to answer the research question. But recently also moved into really big animals, really megafauna, and seeing how we can apply the different models that we have to understand their ecology and also the threats that they face in the changing ocean environments.
Wow, so we could talk about a lot given all of
Yeah,
that you're doing. Let's
yeah.
start by just let our, population ecology or population biology. Perhaps you can let our viewers know a little bit about what that is, because they might not be familiar with it. I have a vague idea.
Yeah. Okay, but so if we so think of a group of animals or plants, they interact together, they survive, they reproduce, so they form a population, so a group of these individuals. And then what we see over time, so over time, those number of individuals in that population will change as a result of, say, differences in survival rates. Maybe some individuals will die, but young ones will grow up and start reproducing. So we see these variations in the numbers of individuals, but also in how they look. Maybe at some periods of time there'll be lots of juveniles, maybe at other periods of time lots of adults. And again, this will determine does the population grow in number or maybe does it decline and how does it respond if the environment is changing, if it gets drier or hotter or wetter.
So we
Yeah.
can use that to sort of understand, like why some species are rare and might be going extinct, and why some suddenly kind of outburst and are pests.
Yeah, it would be a starting point to think about, okay, what is this population and really look at why are some of these individuals or maybe most of these individuals not surviving? Why are they responding to something? Is it, I mean, it could be a disease, it could be a change in the environment. So that is where you would start looking at what's happening to these individuals, to their survival, their growth, and their reproduction, which sort of we use this general term of their life history. So what is their life history from the moment that they're born all the way to they're an adult? What happens in between those two key moments in terms of their survival, growth, and reproduction?
So, Isabel, you said that you use primarily sort of bulb mites
Yeah.
I guess as a model organism. So can you tell us a little bit about bulb mites and what they are and
Yes.
sort of a bit
Yeah.
about their characteristics?
So they're tiny. Let's start there. So the biggest, and the females are bigger than the males. The females at their biggest, they're about 1 millimeter. So you can just about see those with the naked eye, but it becomes more difficult when you look at males, which only tend to be about 0.5 millimeter, and let alone juveniles or eggs. I mean, that becomes tiny. They live in the soil, so they live across the whole world, really. Mites in general, to be honest, live everywhere.
So why are
Uh,
they called bulb mites? I thought they lived in light bulbs or something.
Yeah, they they're called bulb mites because they live on flower bulbs or onions or garlic. So think about your plants that have bulbs, and that's why they live on. And the reason they live there is that these bulbs sometimes they get diseases and then they get fungi that yeah that attack sort of or that eat the bulb, and the mites they eat the fungi. So that's why they're called. So they're bulbs. So they're living on bulbs. They're actually I collect them from my garden. So what I do, I just pick up a flower, I with it that I know as a bulb. I put it out of the garden, and you
Like
look
a
at the
tu like a
bottom.
tulip or a
Yeah,
daffodil or
yeah,
something.
like a tulip. Yeah, or a
And-
daffodil, and you put it under the microscope, and then voila, you see mites.
but if there's no fungus on that
Yeah,
bulb, then there will be no mites either, or will
there
there
will
still be
be.
mites?
Yeah, there will still be mice because they eat lots of detritus and other stuff that's sort of in
Okay.
that small ecosystem. And so going back to if you pull up your bulb from your garden, you put it on the microscope, you also see predatory mites. So it's not just bulb mites living there. There's also predatory mites that again eat the bulb mites. So you've got this really small ecosystem just underneath your bulb in the soil.
And
So,
I mean, of all the animals in the world,
Yeah,
there's like 1.5 million species described. There's like potentially some people say there might be as many as 8 million animals, if you count all
yeah,
the little mites we haven't discovered and the other invertebrates and things that lived in the deep ocean we're yet to find. Why,
yes.
why did you choose bulb mites? Why not like something charismatic?
Yeah, they're certainly not charismatic. Well, well, like I said, so one of the things I'm really interested in is, why do we see this variation in how organisms, I mean, plants, animals, why in how they look? We see differences, and they persist over time. They don't go away. And yeah, if you want to study this in humans, I mean, it'd be quite difficult to put humans in a test tube and do all sorts of manipulations. But with these mites, obviously, it's much easier. So, like I said, they're really, really small. So you can put them in test tubes. They don't live very long. They reproduce after 9 days. They're an adult after 9 days, and so you can study lots of generations. And they showed loads of variation in the way they look. So we've got the males, adult males. They show these two morphs. They're either a fighter. So you have to imagine imagine a round shape. That's a mite, and they've got four pairs of legs sticking out.
So
And
like
then
spiders.
like spiders, yeah, they're in sort of the same broad taxonomy of spiders. And then the third pair of leg of males, the fighter males, is really thick. It's very muscular. They've got a sharp end at the end, and they can use them to stab other mites and basically kill them. And then we also see males that don't have these thick fighter legs, and they're basically defenseless. So we see this variation, and they have, I mean, there's other variation. They have a dispersal morph, which is also fascinating. So during development as a juvenile, they go through this phase where they completely get rid of their whole digestive system, and instead they have this big sucker plate on their belly. And what they do is, so they're in the soil, right? So they crawl out of the soil and they wait for insects to pass, and they just plonk themselves onto an insect with their sucker plate, and off they go to new
So,
horizons.
so they can't feed.
Those can't feed. No,
Huh.
no, they can't. No, so they once they arrive at their destination, wherever that may be, um, again they have to change their whole physiology and morphology. So they further develop. So these are still juveniles. So they then redevelop their whole digestive system and become more of a normal mite.
My God, imagine going on
Yeah.
holiday, Syma, to not eat.
Yeah, exactly. Neither one in
Stuck
your
on,
eye would survive.
yeah, stuck on something.
And so, in general, are there sort of equal populations of the fighters and the scramblers?
No, there's huge variation actually. Yeah, both in labs and outside in the field if you go into your gardens or somewhere else. It really varies. It depends on the food that's available. It depends on different labs. We see different proportion of fighters. Sometimes it can be up to 90% of adult males are fighters, and sometimes none. So really huge variation. And yeah, obviously, why do we see that variation? What's driving all these differences and what's maintaining their coexistence? So those are some of the key questions that people focus on.
So, is it better to be a fighter or a scrambler? I mean, why aren't they all one
Yeah.
or the other? Why is there this kind of variation?
Yeah, so that's a really interesting question. That's the question I started with. Like why do we not see all these only fighters? And you have to remember these fighters and scramblers are adults. So you initially a lot of the theories that biologists use to answer these questions focuses on how do adults perform? Do fighters do they produce more offspring? Do they, fight off other competitors when they try and mate with females? Is that why they're better? So yeah, I worked on that question quite a few years from that point of view. But then later on, none of the experiments actually confirmed any of that. I mean, fighters were not better at getting access to females. Scramblers were equally well able to mate with females. We didn't see any differences. We did not see any fighting. I mean, it's just none of it made sense. And so we actually started reframing the question, more thinking about what developmental process goes, is happens before a male actually develops into a fighter or a scrambler. What happens during development to these juveniles? Why are they making certain decisions? And when are they making these decisions?
And so if you're a
And
juvenile,
yeah,
if I so
cliffhanger.
if I grow up in like
Yeah,
Newcastle or
yeah.
Wolverhampton, am I more likely to be a fighter than a lover? Is that like
Oh
the
yeah. I think you'd be more likely to be a scrambler. So not enough resources. No, so what's one of my PhD students, this is a while back, Floor Rehberger. So he did experiments. He really wanted to pinpoint the moment in an individual's life history, a moment that a male sort of makes that developmental decision to become either a fighter or scrambler. And he found out it is just before they mature as an adult. So really, it's really at the very, very, very last moment that a male will either develop into a fighter or a scrambler. And we think it's to do with stress. So it's to do with stress in terms of resource access, in food, maybe environmental stress. And if a male is in a stressful environment, it might not have the resources to develop these fighter legs, but it will have enough to survive, and then it becomes a scrambler. So the fighter scrambler is more an outcome of the whole developmental process that comes before, and the whole the stress that individuals deal with and the trade-offs that they have to make. So they have limited amounts of energy. Where do they invest it in one aspect or maybe the other? And the outcome of all of that is then that we have males that are fighters and males that are scramblers.
So, Isabel, is this kind of sort of differentiation, sort of late differentiation, I guess,
Yeah,
known in other species?
it does vary. There's other mites, well, there's loads of mite species. There's also other mite species with the same male dimorphism. Um, they haven't unraveled in such detail as exactly where that point is, but we know in other species that that decision takes place quite early. And
Hmm.
again, there's a whole field of research looking into, why do we see these differences in these sensitive windows, so to say, when individuals make these crucial developmental decisions? So it does vary across species. And personally, I think it's got a lot to do with how predictable the environment in terms of food availability or maybe environmental conditions. If it's very predictable, then you can sort of afford to make that decision quite early on.
And then, but you haven't been able to work out what the benefit of being a scrambler or a fighter is. Do you think that's because you're
Mm.
doing it in the lab? And I mean, these things
Yeah.
would be impossible to
Mm.
study in the field. So is there some aspect of the environment that you haven't brought into the lab, the field
Yeah.
environment that you haven't brought into the
Oh yeah,
lab?
no doubt. Yeah. So there's these anecdotal stories of fighters attacking these predatory mites that I talked about earlier.
Ah, okay.
Yeah, and I'm still waiting for a student who wants to tackle that.
Okay.
Yeah. So that's one idea that we have in terms of the advantages of fighters. The other advantage is that if so I mentioned, food shortage can be a real big stressor. If you do manage to develop into a fighter, it means you can kill your conspecific. So the other mites that are with you, at least you have food. So that would be an advantage of a fighter. So a lot of it then focuses on yeah, just surviving, either getting food or fending off predators.
So, Isabel, one thing that
Yeah,
I think, well, Tim and I both get asked often is something that interests listeners and viewers is how do you do what you do? So, Tim
yeah,
said that it's difficult in the wild, in the
yeah.
field. So, in the lab situation, you're looking at can you just talk us through what you do?
Yeah. So I would collect, well, that's what I've done. Collect the mites from my back garden, dig up that bulb, bring it inside, put it under a microscope, and find these individual mites. And then what we use are paintbrushes. So we have a paintbrush. So we cut off most of the hair. So we have a paintbrush with maybe a few hairs sticking out. And then you make them a bit wet. Mites are very sticky, so you can stick them on your paintbrush. Yeah, and then we transfer them into really small tubes. They're about, say, an inch high. And we fill those with plaster of Paris, you know what you use
Yeah.
in, yeah. Because that can contain moisture, because the mice, they do need a high sort of moisture level. So we have this damp plaster of Paris in the tube, and we put our mite in there with, from the paintbrush, and I quickly put a cap on. Mice are very good at escaping, so the cap they need air holes because they do need oxygen.
Yes.
But then underneath that we put a very, very, very fine mesh so they can't escape. And that's how we then keep our mice, and we can then use bigger tubes and have big populations of hundreds and hundreds of mice in there that would happily just live there.
And then you use microscopes to view them.
Yeah, we use microscopes. Yeah, they're called stereoscopic microscopes. So you can put the whole tube under the microscope, and you can paintbrushes. We can't do without paintbrushes because then you use your paintbrush to move mice aside, to pick them up. Sometimes we want eggs. So eggs are like a tenth of a millimeter. Again, you can pick these up with the paintbrush, but then because they're so small, that's also quite interesting. You get the static electricity that plays a really big
Yeah.
role. So they can just boom. So you'd have to be really careful that you're not, you yourself don't have this charge.
Yes.
Yeah. So those are the things that we have to sort of look out for. And yeah, what happens as well. So if you have that setup and you get everything going, what in labs there are also mites live everywhere. You could have mites on your forehead, you notice.
Yeah.
So they also live generally in labs. And what
I'm starting
they
to
do,
itch now.
yeah.
That's so unfair that you.
Well, we've got invader mites, so they're called agar mites. They live in labs and they walk into our cultures, and
Oh.
that's a really big problem. Yeah.
And so you
Yeah.
could then control everything about the environment the mites are in, how much food they get, the amount of water in the
Yeah.
plaster of Paris, the temperature.
Temperature. Yeah, we can tell most of it. But we can't. So some of the things we've seen in our experiments is that even at the finest scale, so you have this small tube, we have hundreds of mites. I mean, it's only a few centimeters high tube. We give them food. We give them yeast, the yeast that you use to make your cakes. They
Oh,
love that
okay.
yeast. Um, but that creates a microenvironment that even if we give them loads of yeast, there will still be mice that don't get enough access to food. So there's even this micro environment, and that's something we cannot control. And I think that that level of variation, so the micro scale variation in a sense, plays a big role in how these individuals develop and whether males become a fighter or a scrambler.
I guess we just can't really imagine what it's like to be that size, even
No,
in a little
it's
test tube.
yeah, it's very difficult. It's I sometimes I show these cultures to people that have never looked through a microscope, and they say, "Wow, that's a whole world down there," which it is.
Yeah.
It is a whole world down there, and it's very difficult to, yeah, put yourself in that position.
So, Isabel, in a recent paper, you've argued that you think these mites have agency, right?
Yeah,
So, can you talk us through that a little bit? Does that
Well,
mean
and you
that
were talking about them deciding to become a
yeah,
fighter or a
yeah.
scrambler.
Okay, yeah. I have to be careful using the word decide. Right. So agency. When biologists talk about agency, well, most of us do not mean consciousness. So we don't mean that this is an individual mind that is having this conscious decision of becoming this or that. They're developmental decisions. They're a result of a developmental process that triggers certain developmental mechanisms as a result of which these males become fighters or scramblers. So agency in this context just means that organisms they actively regulate how they develop rather than passively receiving whatever the environment throws at them. So a mite isn't deciding, I'd like to become a fighter. No, instead it just changes its feeding, maybe energy allocation, timing of maturation and growth, and all those processes together then determine which adult eventually emerges. So yeah, it's about regulation, not about self-awareness. Yeah.
And then, um, so what you talked about stress potentially
Yeah.
playing a role during development. So
Yeah.
does that mean there's sort of some feedback from a stress hormone? Do they have stress hormones like we would? If we get stressed, we end up with lots of cortisol or something like that.
Yeah, they don't have that system. They don't even have such a true hormonal system. They've got these signals, like hormone-like signals. So in insects, for example, juvenile hormone is a big regulator, and mites have some sort of analog in their physiology. Um, I am not aware actually of whether there's a specific one that relates to stress. I mean, there must be a way in which the physiology registers stress because we see them responding to it, and we see these males making decisions. Well, going into certain developmental
Yeah.
trajectories, depending on whether they're in a stressful environment or not.
And it might come down to sort of some of the types of thing that interest Syma in terms of particular
Mm,
molecules going
yeah.
across cell membranes, right? And
yeah,
then, some sort of signal like that.
yeah, targeting a receptor that then
Yes.
starts something else downstream?
Yeah, we have tried actually now that we talk about this, we have tried to figure out is there something that we can block to block
Right.
certain developmental trajectories, and we did find an element, but we were unable to actually, yeah, manipulate that in our lab, so we didn't actually test this. But I think in insects particularly, I think they have done these kinds of experiments.
My God, Syma, we've found a podcast where our interests actually come together through mites of all things.
Exactly, remarkable. And how have you, Isabel, do you
Yeah.
think that sort of climate change and these sort of changes we're seeing are going to have an impact on the behaviors of mites?
Yeah, I think it will, because temperature does play a role in whether males become fighters or scrums. In general, temperature has an effect on the development of mice and all cold-blooded species, because it has the size at which individuals become adult changes if temperature goes up. There's all sorts of impacts on their metabolism from temperature. But we also know about the bulb mite that it affects the fighter expression, so which goes down. So it does seem that the higher the temperature, it's a stressor, and we see more scramblers. Yeah.
So, I mean, do you see the research becoming much more mechanistic in the future in terms of trying to understand development? Because, it's a very complicated
Yeah.
process, and these might start off as an egg, presumably, and then
Yeah.
that egg gets fertilized and they divide into multiple cells, and then some of those cells are going to become legs and heads and
Yeah.
arms, and that's all controlled. It's a really difficult, a complicated process, even in something
It
as simple
is.
as a mite. Is that the way
It
your
is.
kind of research is going at the moment?
Yeah, no, I think for me that's where it stops. I think that I sort of reached the limits of where my expertise is, and time to invest into figuring all of that out. But there's another model system, dung beetles, also very fascinating.
Excellent.
They also show this dimorphism, and they've got all sorts of other interesting aspects to them. And that's where they've done a lot of work, actually, really figuring out the physiology and where in development certain trajectories are switched on and switched off. I wouldn't be able to name it all off the top of my head, but that's a system where they would, yeah, that would be a good one to if you want to figure out more about this and also about agency actually, because these dung beetles, right? So larvae. So you have a mother dung beetle creates this brood ball, so it's a dung ball, and in there she lays an egg. And that's all the food that this particular offspring will have to grow up. And what
Sounds
these
like a
larvae,
shit diet. Sounds like a
yeah,
shit diet to me.
yes,
I can't believe you just said that.
yeah.
I'm on fire today.
But what these larvae do inside is they manipulate the microbiome. So all the microorganisms in that dung ball, so the larvae manipulate that. So they're actually impacting their own development. And that's the type of agency that we're interested in. Yeah.
Oh, that's pretty cool. I didn't know that.
Yeah.
Well,
Yeah.
you get to work with all these sort of glamorous organisms, don't you?
Yeah. Yeah, to be honest, the smaller or yeah, the life cycles just become really fascinating. The number of life stages in juveniles, where they live. I mean, think about dragonflies. If you have a pond in your garden, so you've got these dragonfly larvae eating everything in your pond, and then they have this major transition when they come out and they become an adult. So it's just fascinating.
I'm still waiting to pupate into something beautiful. But but I mean, you mentioned this earlier. One of the really powerful things about such short-lived organisms, you can get through lots of
Yeah,
generations quite
yeah,
quickly because if they're adults by day 9, they're probably dead by day 20 or something, maybe day 30. So you can rattle through generations,
yes,
and you can do experiments. If we bring like lions and tigers into the lab,
yeah,
it's kind of a bit chaotic.
yeah.
They
No,
eat the
that's very
graduate
true.
students. Yeah.
No, that's one of the big advantages that you can do that. I mean, that's the big advantage. The downside is that it becomes difficult to translate to the natural world outside. So that's always the challenge. Like, to what extent do the findings that we see translate, yeah, almost to the real world, so to say. And yeah, there's several ways of tackling that. I mean, one way what I've been doing is to create these fairly simple population models and use the Mite as a system to think about how should we incorporate decisions on energy use and all these things, and then apply them to other species, bigger species, and see
Mm.
how well do they perform. So that would be one way of tackling it.
And because they can, you can get through a generation so quickly, do you sort of start to look at evolution? Because you're talking about different,
Yeah,
you can create populations, say, of different genotypes, I imagine, if you wanted, and you can get all sorts of different individuals, and you can create the populations that you want, and then you can presumably monitor them as they go forward in time.
yes. Yes, definitely. Yeah, so that's actually where so those types of experiments where ecology and evolution meet. So obviously, yeah, as you know, ecology, so when we're looking at changes in the size and the structure of population, has long been one field, and then evolution, when we look at the way organisms look and behave and how that evolves over time, another field. But particularly in these organisms with these short generations time, we can see how both happen at similar time scales. And what's even more interesting, how they influence each other. So if you have an evolutionary response, so say there is a shift in how well individuals are able to cope with food stress, and that's evolving over time, how does that impact the size and the structure of populations? And we've seen that with the bulb mites. So we see if we put them under food stress for quite a while, so say 10, 15 generations, we see that they are adapting to that food stress. So they seem to become more efficient in consuming the limited food that is available. And we even see that that their reproduction starts going up. So females are able to use the energy that they have more efficiently and produce more
So
offspring.
you can see, sorry, Simon, I keep,
Yeah.
so
No, no, go for it.
you see evolution in front of your eyes in animals,
Yes.
right?
Yeah.
And then you can see how that feeds back to influence the population.
Yes. Yeah.
Yeah.
Yeah. And
No, that's a really nice thing again, which is very hard to do with larger animals.
yes. Yeah.
Obviously. So yeah.
And obviously, I guess I'm a physical scientist, so I'm going to
Yeah.
ask this question about reproducibility of results. How do you sort of ensure that what you see is
Yeah.
reproducible?
Well, we replicate a lot. So we think of a particular experiment we want to do. So say, look at the interaction between ecology and evolution under food stress, and then have lots of replicate populations. So different populations that are independent of each other, and we give them the same treatment. And in that sense, we try and reduce that risk that what we see is just some sort of anomaly.
Yes.
Yeah.
And if you took your lab mites that have been in the lab for, I don't know, 10 or 15 generations,
Yeah.
and you put them back in the field, do you think they'd survive, or do you think they've adapted to your
Uh,
laboratory, to your setup?
I think that's might are very difficult to get rid of. Yeah. It's I even had way back I had mites. I just gave them filter paper. So just paper. There's only cellulose in there that the mites
Yes.
can eat. It's really difficult to digest. But they survive. They still survive on filter paper. And they stayed on there for a long time, several years. And what we saw, it was really amazing. After about 2 years, we saw tiny, tiny fighters. So this, yeah, I mean, talk about evolution. So, yeah, we saw they were absolutely tiny, but they were there. That had taken a long time. So that wasn't 10, 15 generations, but probably a hell of a lot more. So more longer term evolution. But it did happen, and we did see those tiny fighters.
I mean, it's remarkable. It's kind of humbling in a way, isn't it, that these tiny creatures, kind of survive and all
Yeah.
thrive in ways that we just simply can't, right?
Yeah, that's true. And you say they're tiny creatures, but they actually also play a role in sort of the bigger process of our atmosphere even. So if you look at mite communities in the soil, it impacts the amounts of CO2, I think. And so if you have, healthy soil, lots of mites, it's good for the environment and for us. So we do connect in some way.
So we're sort of just running out of time, but you're
Yeah.
currently at Newcastle, but it's recently been announced that you're moving down to Royal Holloway. So first of all, congratulations on that. And is this a completely new job, a change in direction?
Yeah, thanks very much. Yeah, so it is a new job. Yeah, so Newcastle, yeah, it's been great in Newcastle. I've been able to come up with, to think about all these ideas and think about agency and where to go next. And I'll definitely miss all the colleagues and students there. But Royal Holloway is it's making a major investment in biology, so they're really growing the biological sciences. And it's also at a time where, many universities are under financial pressure. But for me, it offers an opportunity, yeah, to contribute to this investment. So I'll be the head of the Department of Biological Sciences there, so I can help shape the direction of the department, which I'm really looking forward to work with colleagues there, and to further build all of these ideas by agency and development center research. Yeah, that's what I'm really looking forward to.
No, well, congratulations and good luck
Thank
as
you.
being
Yeah.
head of department, as a recovering head of
Yeah.
department. I think I hope you enjoy it as much as I did.
Hmm.
I did enjoy bits of it. I
Yeah,
did enjoy
bits of it.
it. Any final question, Simon?
I mean, only to say, so are we going to be taken over by little mites then at some point in the future?
Ooh, that's a
Is
good
AI
one.
going to get us first or the mites?
I think the mites will outlive AI and us.
Yeah.
Yes.
Fantastic. Well, thank you very much for joining us today and taking the time to chat. It's been fantastic and really interesting.
Yeah, thank
Yeah,
you very much again
thank
for
you.
having me.
What did you make of that, Syma?
Ah, that was absolutely fascinating. Just this idea of this whole little world of mites that we can't see, or
I know
see
wandering
easily.
around eating tiny bits of fungi and yeast, probably
That's
individual
right.
bacteria. What I really liked about it is it kind of did bring my world kind of down to your world. That sort of,
Yes.
the small animals where and they're where they're eating individual bacteria. But even as we talk, as we mentioned, that molecules going across cell membranes actually driving things like development and the way that these
Yes.
animals develop.
Yeah, and a whole kind of it brings our worlds together, but almost in an untapped kind of way, right? It's a whole new avenues of research in that area, I think, for these cases.
No, I think that's right. And that whole, although Isabel, said she's not getting down into the mechanistic level, perhaps we should get someone on who's a developmental biologist who does this because it's absolutely fascinating. I mean, if you
Yeah.
think, even a mite like that is gonna consist of, hundreds, if not thousands of cells.
Yes.
I think C. elegans, the worm that everyone studies, consists of a fixed number of cells. I can't remember what it is. It's like 139 or something.
Right.
I think it would be really interesting, and maybe we should find a developmental biologist who talks about how molecules actually determine the trajectory that they go in. But I think what I really liked about what Isabel does is the fact she's able to make populations of animals with different characteristics and then see how those populations differ in terms of their behavior.
Yeah, I mean, obviously, as you said, not something we can monitor in humans, but the idea that you can monitor the evolution of populations in real time is mind-blowing, really.
No, it is. But I still, despite that, I still plan to go out to exotic locations to look for wolves rather than going into my garden, pulling up an onion or a tulip.
Or looking through poop for dung
Yeah.
beetles.
But you do need, I mean, I think you need all of these different, and I think all of these different approaches. And I think that's why science is so incredible, right? You've got all of these different approaches that come together.
Yeah, absolutely. And it
Yeah.
keeps, and also it keeps us in work and keeps us being able to produce this wonderful podcast.
No, it does. And I mean, hats off to her as well, because although, she's very modest, this job she's got as head of department, that's obviously going to be a big role. And it is nice that the universities are still able to invest, or some of them are invested in
Yeah.
kind of our sort of areas, because it's pretty tough out there for a lot of universities at the moment.
Yeah, absolutely. And this the thing is the work is vital and it's going to shape the way we do things, the way we understand the world around us and this, as you said, investment in these areas is absolutely crucial and yeah, it's encouraging to see that some areas, some places, some institutions are still doing it.
I know, particularly given, there's news this week that Exeter is cutting a significant number of posts, for example. And, a
Without
shout
being
out,
under
I've got.
financial pressure,
Yeah, it's
right?
financial pressure. I mean, it's all down to the way that universities make money, which is the number of students that come in, and it's a competitive space. If you're not able to bring in enough students, the government funding for universities hasn't risen for some time, and it's a really difficult one, right? Because you don't want to burden students coming in with significant amounts of debt. They still have to take loans to come to university, many of them. And so the finances of them just don't add up. So many of them are cutting areas. And I think it's a difficult decision for universities to make, but and we always think they're making the wrong ones, of course. But so, some of my colleagues at Exeter in psychology, I know, at threat of losing their jobs, and also across the humanities, there's a significant loss. And I really feel for them, but it's difficult. So So this is becoming a political podcast. It's almost,
Yeah,
hopefully we'll get listening figures up there with the rest is politics now.
that's right. Come and listen to us. We're better.
Yeah, no, exactly. So excellent. Well, I will report back. We'll be online again. In a month or so. We're kind of
Yes.
reducing the frequency, but I can tell you what it will be like up in London, and by then I will have been back from a trip to the Galapagos.
Oh, wonderful. Yeah, so look forward to hearing about that. So yeah, I think actually I think maybe a podcast on the trips on the Galapagos would be quite interesting.
Or I'm sure we can find someone who works on the Galapagos to get on.
Yeah, I
Absolutely,
think
we
so.
can hear about that. Yeah, good idea.
Perfect. All
Good
right,
idea.
great. Thanks, Tim. We'll see you when you get back.
Yeah, have a good one. Okay, talk to you soon, Syma.